Thanks, Emily. Thank you for the invitation. This is great. Okay, so I'm going to give this talk, but I'm going to share with you the capability of remote sensing as a tool to be able to monitor this marsh ecosystem more effectively. I don't have any GOMRI funding now, so my GOMRI funding ended in 2012. But I have been getting funding from NSF and NASA to be able to continue this research. All the results I'll be, for the oil spill related work will be up to 2012. Then I'll basically talk about a more general remote sensing based approach to map or monitor marsh biophysical parameters. Okay. I'm from University of Georgia. I manage the remote sensing and field spectroscopy lab over there. So before I go into the talk, I just want to give you a quick pitch about remote sensing. Why we use remote sensing? What's the idea? I think you might have seen this before. It's basically cost effective. So you can monitor a particular patch of wetland or particular area frequently, every week, every month, daily. We have satellites in space that does daily twice, once in the morning, once in the afternoon. So that increases your chances of getting cloud-free, good quality data for frequent monitoring. Then so that's the idea of multi-temporal coverage. And then it can make inferences about inaccessible areas. Okay, so those are those things that I think we know. The beauty of remote sensing. It's not an exact science. I just want to be clear. Not like Brian's biogeochemistry. 15-20% error in doing things from space is pretty okay. I do a little bit different types of remote sensing. So basically combining field and lab-based spectroscopy, which is basically remote sensing from few inches. Remote sensing is, as long as you're not touching the target, you're doing remote sensing. So few inches, few feet, you get controlled data set, and then you scale it up to satellite level measurements. The process of field spectroscopy, we call it proximal sensing. It's basically your close proximity to your target. The biggest source of error in remote sensing data set is the atmosphere. Okay, somebody's noise is somebody's signal, right? So our noise is atmosphere, which is signal to atmospheric scientists. What atmosphere does is add a lot of extraneous scattering to the remote sensing signal. So your spectra or spectral signature from the target get really diluted when it goes through this two-way traffic of sunlight coming through the atmosphere, hitting the target, and going back into space again to the satellite, right? So by doing field spectroscopy or proximal sensing, you can actually examine the interaction of EMR, which is electromagnetic radiation, with the target and develop spectral models. Those spectral models can be then upscaled to whether it's drone or aircraft or satellite-based altitude. And it also helps us validate the satellite maps, products. We don't want to send out a map without accuracy number attached to it. So these field measurements will let us do that. So this is one of those sampling photographs in Marsh Point, Mississippi, during the height of the oil spill. So we go there and we calibrate our sensor with a known reflectance panel, and then we put it up, and then we start collecting data from different marshes, different degree of oiling, whenever we got access to the oiling site. Most of the time we didn't, but that's the idea. A little backstory. I joined as a graduate student in University of Nebraska, Lincoln. So first couple of years I did remote sensing of corn, soybean. So that's a really fancy platform with a lot of sensors hanging out and basically looking at the yield or whatever of corn. I got really bored and I got a complete U-turn to remote sensing underwater. So I did a lot of National Geographic style work, scuba diving and looking at remote sensing of coral. I kept the letter C from corn to coral. So this is also remote sensing. You can see we put a tripod really close to the coral. This is Roatan Island, Honduras. So the tripod, the sensor is not touching the coral. It's very close to the coral. So we wanted to see the photosynthetic activity in the coral for 8 hours. Okay, from the sun going from covering the so different degree of sun angle. So we did a lot of experiments. We are from Nebraska, so we didn't have any marine science background. But that's one of that's chapter 4 of my dissertation, where I call this instrument ABD, all but defense. So this is basically a buoy system. With few sensors looking up, few sensors looking down, and the idea is if all these divers will release the rope at the same time, and the rope has markings, then the whole system moves up and it starts to see more and more coral. And so seeing more and more coral will give us a better representation at the satellite level, okay? It's very difficult to match point measurement with a pixel. So you have to have a footprint, which is representative of the satellite pixel, okay? So then I got a job in Southeast, and I didn't have corn, neither coral, so I reached a compromise, and I'll do wetlands, it's in between land and water. So that's what we do now, remote sensing wetlands in various ways. Okay, so now going back to the actual wetland monitoring. We know the ecological, economical value of wetlands, from nutrient trapping to storing large amount of carbon to providing fresh water, flood water reduction, attenuating storm surge. So we know all that background information. You might have seen this kind of photographs in websites. They show the degree of defragmentation in the wetlands from 1800 to 1993 in this example. But you might have seen the USGS projections, which is up to 2050. So that is 2050. The marshes that are prone to be lost, and where are they located in terms of the Louisiana coastline. So we're aware of the land loss problem in Louisiana. I mean, there are lots of statistics, mainly happening because of engineering modifications to the landscape, to relative sea level rise, climate change induced. So my idea was to go beyond the land loss, land gain, to something a little more ecological, which is basically looking at the biophysical properties, which basically tells about the photosynthetic capacity, the physiological status of these wetlands over the years. So, we know about the BP oil spill, approximately 206 million gallons of crude oil, the first major US oil spill to affect marsh in that extent. And so the immediate impact, as predicted, loss in green biomass. So a lot of browning happened. Reduction in photosynthetic capacity. Oil coats the leaf and canopy, and then reduces the photosynthetic activity, thus turning the marsh yellow in the middle of the growing season. And then impact of the cleaning effort for burning, flushing, and skimming. So these are the photographs taken in the field in 2011, 10, and some in 11. So right after the spill, I got funded from NSF and GOMRI, the bridge funds, to basically continue this work, because I think we are one of those groups who got funded in 2009 before the spill to do this exact same work from NASA. So we have been collecting field data since 2009, so basically to keep it going. So the idea was to look at 4 parameters. Those are biophysical parameters that can be mapped from space, and they tell a lot about the marsh, other than just area loss, area gain. Those 4 parameters are canopy chlorophyll content, the total amount of chlorophyll A, which drives the photosynthesis, then green biomass, which is above ground, anything green, okay, weight per area, and then leaf area index, which is amount of leaf exposed to the sunlight. So that means that directly affect the interception of light to the leaf, which drives the photosynthesis. And then green vegetation fraction. So basically, how much green, how much everything else, brown or yellow or whatever. to use remote sensing data to develop models and then scale up those models to be able to map it and map it every week since 2000 to 2015. So we're looking at 5,000, 6,000 maps for the entire Gulf Coast. So you can click on a pixel, any area, and you get a really high frequency construction weekly for 15 years to see the signatures of hurricanes, tropical storms, prolonged drought, oil spill, and whatever else is happening in that region or that patch which you're experiencing over the years. So the idea is that these products, can identify hotspots, can basically, coastal resource managers can use it to identify candidates which are, prioritized for restoration projects, things like that. Okay, so the field method works very simple. It's basically incoming sunlight and how much light is being reflected from the target. In this case, it's marsh canopy, right? Basically radiance over irradiance, so we use a dual sensor approach. If you use a single sensor that is looking down the marsh, then every time it becomes cloudy or shaded, then you have to recalibrate your sensor. So dual sensor approach, what it does, it basically gives you relative reflectance. Anytime it becomes cloudy, the reflectance goes down, so it gets normalized. This is the frame we used. We tested a lot of frames. This is the frame we use because this is the frame that gives us the footprint of 2.2 meter diameter. So IFOV is the instantaneous field of view of the sensor. That means how much area it's looking at. It's always look at a circle, and that is a 2.2 meter diameter. So if you can take enough reading of 2.2 meter readings on a marsh patch, and then you average them out, then they might represent a satellite pixel more accurately. Okay? Taking one reading might not do the job, so you have to optimize and you have to find out what is the representative number of readings in a particular pixel, whether it is Landsat or MODIS or any other sensor. So it will depend on the spatial resolution of the sensor, which is the pixel size. So we calibrate the sensor. The sensor has a laser pointer, so it will always point to the middle of the scan. Then we have the PVC pipe that represents the total, scan area, and then we do cookie cutting to find out the exact area that the sensor scanned. And you do that enough so that until you get a close match with your satellite data in terms of pixel reflectance pattern. Okay. Basically we use multiple sensors. Some of the sensors are really light and tiny, so they can be mounted on this frame like this and look at a 2.2 meter diameter area. But some of the sensors are backpack systems, which are much heavier, like 50 pounds or so they cannot be mounted, but they're very, very accurate. So we also take multiple scans with that within the field of view, just to make sure that there is no outlier or something like that. So overall, before and during and after the oil spill, we collected lots and lots. I call it spaghetti plots, a lot of spectra. And the objective was to basically let's develop two types of model. One is spectral model for each species individually, because when you are looking at the marsh from such a close distance, they have a lot of species signature. Okay, so the reflectance changes because NIR light, which is totally controlled by the leaf structure, canopy architecture. It's nothing to do with pigments. Okay, only the visible light, they get absorbed by the pigment for photosynthesis, but NIR gets scattered around based on the leaf area and other structure. So, the species signature is very, very strong in situ data, which is proximal sensing data. But as you go up in space, up in the altitude, the species signature get diluted. So no matter how much variability in terms of canopy structure in marsh grass, the satellite reflectance doesn't change, which is a blessing in disguise to be able to do a species invariant model, which is not sensitive to changing species. Of course, if you find a big patch of juncus, which is really dark compared to Spartina, then there will be some differences. But overall, that species variability is lost basically with altitude. So whenever you do this remote sensing data collection, you have to do a lot of biophysical in situ data collection to be able to calibrate and validate your models. So this is, for example, the leaf chlorophyll content. Leaf chlorophyll can be transformed to canopy chlorophyll depending on the area of the leaf and stuff like that. We do LAI readings, which is leaf area index. So leaf area index reading is pretty simple. You take a couple of readings from the top of the canopy and a bunch of readings from different angles in the bottom of the canopy, and the amount of sunlight that is lost from the top of the canopy to the bottom of the canopy is basically used in something which is photosynthesis. And so that's how you convert the FPAR, which is fraction of photosynthetically active radiation, into leaf area index. This is the Veg Fraction I was talking about. Veg Fraction is that frame has a digital camera on it. So it will look at the exact area that got scanned, and then that area will be converted to Veg Fraction. So basically it's a pixel counting, simple MATLAB code that counts how many green pixels and how many everything else. So in this case it's 86%, okay? In this case it's 42% and so on. So every spectra we collected, every remote sensing data we collected had all these biophysical parameters associated with them for model validation, calibration. We did biomass destructive sampling and then separating the green versus brown and drying the green, weighing the green, and all those things. So basically, that's the kind of data we got. So this is taken from the frame, which is looking at two different, marsh, footprint, and then two different spectra, saying something about the footprint. More area of water exposed, then the NIR goes down. More marsh exposed, NIR goes up, and so on. We saw many uninvited guests in our during field trips. So I have tons and tons of pictures of creatures than spectra. Okay, so in oil spill, during oil spill time, we didn't have access to the field most of the time. So what we'll do is, really early in the morning, around 7, we'll charter a single engine plane and go look around where are the yellow tapes, because they'll put yellow tapes in those areas they cleaned up, and we basically, kind of roughly record the GPS readings and try to go to those sites. And we went to a bunch of those sites, and marsh impact was obvious. In some cases, they're totally coated with the oil. Some cases, the root structure is affected by the oiling. And here we're taking biomass sample, and you can see sign of oil all over in those marshes. So here's the spectral signature in terms of magnitude of oiling. So interesting thing we noticed that between healthy marsh and moderately oiled and severely oiled marsh is that the sign of a healthy vegetation is the strong absorption of light at 675 nanometer, which is basically the characteristic chlorophyll A absorption feature. So if the marsh has got a lot of oiling in it, that means it's under stress, that means it's not photosynthesizing that effectively, then the 675 nanometer absorption will start reflecting more and more light, okay? So the less prominent the 675 nanometer absorption feature, that means more contamination or more oiling. And that was pretty obvious in the field data we collected. Looking at I'm not going to go into detail on these modeling aspects, but looking at the models, you see a strong species signature in the in situ models. So different species: Batis, Disticulus, Juncus, Salicon, Spartina, Alterniflora, patens. So they have different performance in terms of simple biomass model in this case. But that performance is that those lines actually come together when you look at satellite data and start modeling from the satellite altitude. Different species, different correlation in situ data, which is taken from the frame, which is 16 feet altitude. So that was the field component. So in 2012, I just wanted to see the short-term quantitative estimate of oil impact. So basically a change detection project between 2009, 10, and 11 to see what has happened. And that was published in Remote Sensing Environment. And these are those Landsat data we could get. So you have to be careful with the Landsat in terms of, because they're 16 days temporal resolution. So So there is a lot of tide. You have to look at the tide. You have to look at the cloud cover. So getting common area Landsat for a large-scale change detection project is very, very difficult, because not all areas are going to be equally clouded. But you need, so the first rule of change detection, you have to have a common minimum area between these images so that you can do the change algorithm. So those are our 2009 and 2010 Landsat dates. Pretty close. So another rule of change detection is that you have to have anniversary dates or close to that. So we got pretty close to that in terms of monthly change. The other aspect of satellite remote sensing is to remove the atmospheric signal, which is by itself is a big field in remote sensing, to remove the atmospheric scattering and absorption by atmospheric gases, the aerosols, the water vapor. So we did a model on, it's called FLASH atmospheric model. So you can see the comparison between Landsat data, which is the solid line, and then the proximal sensing data. So if this step goes wrong, then all the rest of the modeling is basically wrong. So you have to come really close to the in situ level to be able to run these models. Anyway, so we ran a lot of vegetation models. On top of that, marsh is a pretty difficult environment to model on because of mainly water impact. So what happens is that when there is a satellite is looking at a lot of water background, the NIR is trying to absorb, because NIR gets absorbed almost 100% in water, but NIR get reflected a lot in vegetation. So NIR is basically struggling between water and vegetation, whether to reflect or to absorb, and so it's basically not that prominent like terrestrial grasses, but it's not that subdued either. So it's basically in the 20-25%. So some of these models we got decent results, and we just went with that. And here's the validation. Validation is percent normalized RMSE is around, highest error was 12% in this Landsat modeling study. So if you take that model and apply it to your 2009 image and your 2010 image and perform a simple change detection, this is what we got. We got a lot of red pixels, but we wanted to have a really conservative estimate, compensating for all the errors, which is highest is 12%. So we basically said, okay, show us all the areas which are experiencing a decrease in canopy chlorophyll greater than 20 milligram per square meter. Okay, don't show us any other, any area that is experiencing decrease less than 20. Okay, give us the real conservative estimate. So those are the pixels that got highlighted that experienced a decrease within that year from 2009 to 2010 of at least more than 20 milligram per square meter. In terms of biomass, very similar, very corresponding change. But those marsh patches that got experienced lower chlorophyll content also decreased in green biomass. Okay, so that was the first immediate quantitative assessment. We also looked at multiple parameters. You can read the paper. It's we'll look at rainfall, we'll look at temperature, we'll look at PDSI, which is drought index, and everything. 2009 and 2010 were pretty comparable year. Not any hurricanes in either years. We can safely isolate this is the spill impact on those marsh patches, okay, within a year. Then in 2011, we noticed that most of these patches are back to the 2009 level in terms of above ground canopy chlorophyll or biomass, all those properties. So I cannot tell you that this continued in 2011. It was slightly less red pixels, not like this much, but there are only few patches remained in 2011 that continued experiencing decrease in canopy chlorophyll or green biomass or the other two biophysical parameters. So the decrease in area, how much area experienced that decrease? In 2009, it is like 50 square kilometer, which is attributed to the natural variability in the marsh and some patches. But in 2010, it was a significant increase up to 420 square kilometer area that experienced that decrease in the middle of the growing season. Okay, so these models are built for growing season only from April to October. And in the middle of the growing season, they, 450 square kilometer area is experiencing either a decrease of 20% in canopy chlorophyll or green biomass. So we, looked at few, pixels of marshes, fringing marshes and interior marshes. We saw some interesting trend, and that trend actually didn't change that much from pixel to pixel to pixel. In the fringing marshes, you can see the immediate impact of the spill. So this is the phenology. So how the growing season, should be like a sine curve, peak growing season August, September, and then slowly declining. But in the immediate impact in the fringing marshes, you can see in June. And then start to recovering some of those patches. Maybe they got cleared, they got cleaned, and in interior marshes there's a time lag. So you can see the decline in August, late July and August. Looking at some of the other modeling results, it made sense that oil took time to get into those intertidal waterways to affect the interior marshes. And the similar results was seen in another satellite, which is MODIS. MODIS is the satellite that covers the area daily twice. Once in the morning around 10 a.m. MODIS Terra, and once in the afternoon at 2:15 MODIS Aqua. So you can see the similar response of MODIS derived LAI. MODIS is a little bit coarser resolution pixel. It's 250 meter by 250 meter. So you're getting a lot of averaging out effect in MODIS. Now I will. So since then, I realized that we need to do a long-term analysis of these marsh habitats, and the only way you can do it is not by using Landsat, which is pretty patchy. There is cloud cover. There is every 16 days. If it is cloudy day, happens to be on the 16th day, then we get a really cloudy image. So MODIS is the way to go because of the temporal frequency, and MODIS provide a 8-day cloud-free image. So it's surface reflectance, which is downloadable for free, 250 meter and also 500 meter. Okay. So that project was implemented in Georgia, similar marsh gradient. Mainly Spartina dominated ecosystem, basically collecting similar in situ data. That time I moved to Georgia, so it was hard to come back to Louisiana to do field work. Collecting ground data, collecting NWI, which is National Wetland Inventory, wetland areas, cropping the satellite data, then running the model, then weekly, monthly map composites, and then doing a post-time series analysis of phenology, or weekly and monthly maps. So basically phenology like that and maps like that. Okay, so that is possible using MODIS. And here's the animation that shows weekly distribution, spatio-temporal distribution of any biophysical parameters, those 4 or 5 biophysical parameters that you can do using MODIS data. Okay, so this is showing the growing season, then senescence, whatever is happening in Georgia coast. So similar model, can be implemented to Louisiana coast or Gulf Coast. I'll show you some of that result, and you get a lot of information. It's kind of big data project where you can analyze a patch for 15 years, because MODIS is available since 2000. It was launched in 2000, so 2000 to 2015. We did some other interesting analysis. We overlaid those phenology with drought. Georgia affected by drought quite a bit. So these yellow boxes are the drought years, and every time there is a drought year, you can see the overall biophysical parameters went down. Not very strong correlation, but significant one with PDSI for those dieback sites. For example, Georgia has dieback sites that is managed by the DNR. People say a bunch of reasons affecting the dieback. One of the reasons is maybe drought, and this is the correlation of dieback sites with drought. It's significant. So you can actually do an overall trend analysis of a particular marsh patch to see how they are behaving in terms of in last 15 years. Whether you can compute something called phenometrics, the length of the growing season every year, or the peak of the growing season. Is the peak of the growing season moving to September or October or moving back. So you can do a lot of analysis from these phenological data from models. So we have those data sets available for you to analyze. You can give me lat long of your site, I can give you a phenology for 15 years. Okay, so these are the maps. For example, I'll show you, these are the maps we have in our archive. This is pre-Hurricane Gustav, and this is post-Hurricane Gustav. Okay, so hurricane impacts are the most severe on those marshes, immediate signature change in the marshes. This is post-Katrina, pre-Katrina, this is post-Katrina. So you can actually overlay the track of the hurricane, and you can see you can analyze the strongest side of the hurricane, and then you see the impact after you get a cloud-free image the very next 8th day or 16th day. So if you zoom in into those map products, you see a lot more detail. For example, you can put a hurricane track and see zoomed in levels of pre-post hurricane analysis. You can do same with Katrina. A lot more severe impact. So here is how it happens. So you have this 6,000 maps in a database, and you can take your lat long, or you can take your area, of whether it is a restoration site or whatever, and you can construct these phenological plots. Of course, these are an average of multiple pixels, but you can then overlay your so these data sets, this drought is downloaded from the National Drought Mitigation Center. These are the hurricane signature. You can see Hurricane Isaac here, slight dip, and you can zoom in on those phenology plots to a year or 2 years and look at it in more detail. This is for 250 meter data, and this is for 500 meter data for the entire Gulf Coast, South Marshes. Okay, so conservation, restoration implications. Personally, I like to see analyze more the effectiveness of restoration projects. We have a lot of restoration projects going on in the Gulf Coast in Louisiana, so we can do a pre-post restoration analysis, or you can do a trend whether it has actually improved in terms of photosynthetic activity, for physiological stress, whether it has improved, restoration has improved the marsh health or productivity trend. So this methodology facilitates prioritization of conservation and restoration efforts, predictive ability to, so this is where we are headed now in terms of research, predictive capability with respect to carbon sequestration in these ecosystems. Okay, so carbon sequestration literature, if you scan through those, a lot of them link to vegetation index like NDVI and stuff like that. Vegetation index for wetlands, they don't work that great. So my argument is, if you replace this vegetation index with biophysical parameters like LAI, then it will link very well to productivity. So take these biophysical parameters and then link it to CO2 flux. Okay. So this is our ongoing project right now in Louisiana, Mississippi coast, funded by NASA ROSES, is to look at the carbon sequestration potential of these marshes by installing flux towers and taking long-term data for 2 years, every 30 minutes. So we want to be able to answer questions like which species more efficiently sequester carbon, what happened to the carbon sequestration potential of wetlands after natural anthropogenic disasters. We will be able to tell you in terms of carbon, not in terms of area loss, area gain, but in terms of carbon loss to the atmosphere. Okay, what is the effectiveness of a particular restoration project? So we, this project is already ongoing. We have built the boardwalk, one in Grand Bay, one in by Sauvage in Louisiana. So you can see that's the flux tower. It gives us data every half an hour, so CO2 flux. So basically you take the net ecosystem exchange and you subtract the nighttime respiration, you get the gross primary productivity of the marshes. And then you can, the idea is to be able to create a GPP spatiotemporal map to be able to say carbon sink, carbon source, which areas are hotspots or going through degradation. And so we do a lot of drone work now, recreation. We're not doing it for research of those sites, and this is going to get more involved with more newer, cheaper, and lighter sensors coming into the market. So it'll be a platform in between our satellite and in situ sensors. So I'm a big proponent of reproducible research. So everything we do in the lab is documented in the website, and we produce a detailed guidance manual for each of those models. So all you need to do, because all this satellite data is taxpayer, free data. Anybody can download. You don't even need a login ID or password. So you can download the guidance manual, starting from unzipping the files to producing the maps. So everything is in the guidance manual that anybody can, have a free access to software like QGIS can be able to, reproduce. So a lot of people to thank, a lot of agencies funded this work and funding currently in this work, and that's it. Thank you very much. I'll take questions, but before that, I want to run something in the background. Now you can ask questions. This is a drone image, drone movie. One of those marsh patches. You can see the drone starts running. Now it will take off. It's very stable. It gives us. We use it to basically look at alligator nests before we start walking in the marsh. but this can be used. So this is from our drone, which is super stable. Now we have techniques, photogrammetric techniques, where you can take this video and extract photographs every fraction of a second and create a 3D model of the marsh, which basically can be a proxy for the standing biomass. Okay, so that technique is called structure from motion, and it's really popular nowadays. So maybe next time I can show you some biomass approximation from the drone. Thank you.
We take 2 questions for Dr. Mishra.
Can you do LiDAR on the drone?
Yes. So the idea is to do LiDAR type of products from the drone. So the idea is not to take vertical photographs, but a low oblique photograph, and there has to be.
They may not be up here.
Oh. Yeah. So you can do exactly LiDAR type of point clouds from these drones, but there are a couple of rules. You have to have at least 60 to 70% overlap between photographs. So it's always recommended to take video instead of photograph because you get more overlap, and then you extract every 0.05 seconds or whatever, and then create a point cloud. But another rule is you have to have a little oblique photograph, like 75 degree instead of 90, so that you can see the side. And if there is a lot of texture in the photograph, then you can create a point cloud. And I have tons of examples. I teach a class on structure from motion, where people, students just go out, take digital photographs, and make 3D models. And there are a lot of softwares out there that can actually do that. So, yeah, for sure. And these drones are exploding right now because now we have a sensor which is like our in situ hyperspectral sensor that is imaging, and that can be mounted on the drone. The only classified information is the inertial navigation system, where you upload a flight path and tell the drone to go from point A to point B without any help. So that is the only thing that is classified, but everything else taking hyperspectral data, putting it in a differential GPS to georeference the data is.
Can you see the bottom, or only the canopy?
I mean, it depends on the camera, and depends on the water quality. But if the water is clear and you have a blue band, then you should be able to see the water.
One more question.
You have to ask one more question.
How far into the future do you think we'll continue to see the effects from the oil spill?
Well, the effect of the oil spill on the above ground, photosynthetic activity in terms of biophysical parameters we're not seeing it since 2011. That was the immediate short-term impact, but it depends on what is the effect of the degradation of the oil in the soil, and what's its impact on the wetlands productivity. But so far, the above ground stuff is not showing any sign. So the idea is to keep on monitoring to see whether it can be isolated.